Hello, good morning and welcome to all of you. I am Rajesh Kumar Singhal and I am here to present a new lecture on the wheat crop, the past, present and the future of the wheat crop. So friend, this is a very important topic for all the agriculture students, and this topic is also important for all the competitive exams related to the agriculture. So friend, in this lecture we will discuss the different facts of the wheat crop. So we will start our lecture with the some basic points of the wheat crop. Then we will discuss about the botany of the wheat crop. Then we will discuss about the prominent species of the wheat crop. We will also discuss about the important growth phases of the wheat crop. Then we will discuss about the some important traits like plant height. We will discuss about the crop phenology, how dry matter partitioning and yield components, some yield attributes, then yield improvement. We will discuss about the role of canopy temperature depreciation. We will discuss about the carbon isotopic discrimination. We will discuss about the use of NDVI sensor. We will discuss about the some breeding, biotechnological and physiological approaches. So friends, this topic is very important and we will discuss about the physiological, biotechnological and the breeding approaches. So let's start that our discussion with the wheat. So wheat is a grass. Okay. So friends, you know that mostly all the grasses belong to the Poaceae family, and wheat is also belong to the Poaceae or Gramineae family, and most of the crop from this family is belong to the C4 family, but this wheat crop is a C3 grass, and this wheat is widely cultivated for the seed purposes. Okay. Second point is that wheat is a major staple food around the world. Okay. So millions people worldwide depend upon the wheat crop for their staple food or the staple energy. Okay. So this wheat crop is also belong to the genus Triticum. Okay. The most widely grown species is the Triticum acetivum Ag. Okay. This is known as common wheat. Okay. क्योंकि सबसे ज्यादा ग्रो करती है इसलिए इसका नाम क्या पड़ गया, कॉमन wheat. And wheat is belong to the Triticum genus and species is the Triticum acetivum, which is widely grown. If we talk about the temperature, the optimum growing temperature is about 25 degrees centigrade. That is mostly room temperature. Okay. With a minimum of 3 to 4 days. Okay. And maximum is 30 to 32 degree. Okay. Wheat kernel is Caryopsis type. Okay. So this is a wheat grain. This is Caryopsis type. Okay. And the protein content in the wheat grain is approximately 13%. Okay. तो प्रोटीन कंटेंट कितना होता है, approximately 13%. And the major protein is the gluten. Okay. That is used is multifarious in the food industry, like making of the bread. Okay. And gluten can trigger some allergic responses in, okay, like celiac disease, non-celiac disease, some gluten sensitivity, gluten ataxia, dermatitis herpetiformis. When we use this gluten in the higher quantity or higher amount, some person are sensitive to this gluten, and this can cause some celiac disease, non-celiac gluten sensitivity, gluten ataxia, and dermatitis herpetiformis. Okay. So this is the wheat crop field, and these are the top 3 countries which are producing the wheat, top growing wheat and top producing wheat, that is China, India and the Russia. Okay, they are producing more than 100 millions of ton. Okay, so now comes to the botany of the wheat crop. Okay, so the wheat crop height is medium to tall, that is 70 to 120 centimeter. Okay, then stem is jointed. Okay, so these are the stem of the wheat, that is jointed. Okay, and usually hollow. Okay, so wheat have hollow jointed stem, and sometimes many stem in plant, that is known as multi-tiller in the field conditions. Okay, this wheat have narrow leaves. Okay, like this. Okay, so their base, this is the leaf base, sheathing the stem one above each. Okay, like this. Okay, so they are covering the stem, that is known as leaf sheath. Okay, and at the top of the stem is known as flower bud. Okay, flower head, sorry, this is known as flower head. Okay, containing 20 to 100 flowers. Okay, and each flowers contain both male and female parts. Okay, तो अभी तक हमने क्या पढ़ा, एक तो 70 to 120 सेंटीमीटर, stem is jointed, usually hollow, and many stem in one plant, sometimes multi-tillers, long narrow leaves, and their base sheathing the stems, means they are covering the stem or protecting the stems one above each joints. At the top of stem is the flower head, this is flower head, it contains the 20 to 100 flowers, and this each flowers contain both male and the female flowers. Okay, अगर मेल और फीमेल फ्लावर्स है, मिस मोस्टली सेल्फ पॉलिनेशन इज टेक प्लेस. Okay, so the flowers are wind pollinated, while more than 99% of pollination events are self pollinated. Only 1% is the wind pollinated. Okay, the flower is housed in a pair of small leaf-like blooms. So this is blooms. Okay, this is blooms. Okay, the flower grouped into the spikelets. Okay, so friend, this is a diagram of one spikelet. Okay, each with between 2 to 6 flower. Okay, each fertilized carpel develop into the wheat grains. So 2 to 3 wheat grains are jointed. Okay, botanically this fruit is known as caryopsis, or berry is also known as. Okay, also called as a seed. Okay, so the grain ripen to a golden yellow. तो जो आपका wheat है उसका कलर कैसा होगा, wheat grain का golden yellow, and a head of grain is known as ear head. So this is known as ear head. Okay, so these two are the lemma, and this is palea, and this is full spikelet. Okay, so this is about the botany. Also, leaves are emerged from the stem, that is known as shoot, apical, meristem. Okay, in a telescopic फैशन. टेलीस्कोप कैसे निकलता है, ऐसे. तो इस फैशन में आपकी रूट निकलती है. ठीक है, दिस इज द डेवलपमेंट ऑफ टेलीस्कोपिक फैशन. ओके, The last leaf produced by a wheat plant is known as flag leaf. So friend, this is also one of the important leaf and the major leaf which provide the photosynthesis during the later stages. ओके, जो आपकी रिप्रोडक्टिव स्टेज है सीरियल्स में, उसमें फ्लैग लीफ हैव इंपॉर्टेंट रोल इन प्रोवाइडिंग photosynthesis to the growing grains. Okay, so this have denser and has a higher photosynthetic rate than other leaves. So these leaves have higher photosynthesis, pigments and the higher photosynthetic rates, and it's provide carbohydrate to the developing ear. Okay, so this is the role of the flag leaf. Also, wheat is a unusual among plant having more stomata on the upper surface. जैसे आपकी ये leaf है, this is upper surface, that is known as abaxial sides of the leaf, then the under sides, that is known as adaxial surface. Okay, upper is abaxial surface. The shoot is made up of a series of repeating units और फाइटोमर्स. ओके, तो ये आपका जैसे वीट का स्टेम है, इसमें रिपीटिंग यूनिट होगी. ओके, सो दिस इज नोन एज नोड्स. बिटवीन टू नोड्स द स्पेस इज नोन एज इंटरनोड. ओके, एवरी नोड देयर इज ए लीफ. So one phytomer have one node, okay, one leaf and one elongated internode and one bud in the axial of the leaf. Okay, so this is known as phytomers. Okay, अब बात करते हैं रूट की. So wheat root are among the deepest of the arable crops. It can extend up to 2 meters. तो जो आपकी wheat root है, that can go up to 2 meter in the soils, and there are two types of root are found, that is primary or secondary roots, or you can say seminal and the nodal roots, or primary or the adventitious roots. So these are the crown root which arise from the lower nodes of the shoots, means they are arising from the, and adventitious root arise from the primary root. Okay, wheat may be on or not on. So this structure is known as on. Okay, producing awns incur a cost in grain number. Okay, अगर आपके awns ज्यादा होंगे, then there will be a less grain number, but these awns provide some photosynthesis and are able to photosynthesize when they are green and more efficiently than their leaves regard to the water use. Means they are more water use efficient than the leaves. Okay, so friend, this is about the botany, that wheat plant is a typical grass having the hollow stems, having the nodes, internodes, then cylindrical or narrow leaves arose in the telescopic fashion. Okay, and at the leaf base they have leaf sheath that is covering the stem, or that is also known as protecting sheath, and also they have upper flag leaf or last leaf that is known as flag leaf that provide photosynthetic to the growing ear heads. Also they have more stomata on the upper side than lower sides. Okay, so we can say that is less water use efficient. Also they have two types of root system. They have seminal and nodal roots, okay, or primary or secondary roots, and they also have on or not on leaves. Okay, so this is about the botany of the wheat crop. Now come to the major species. Okay, so in the wheat there are three types of species. The first one is the hexaploid, that is 6n, tetraploid, that is known as 4n, and diploid, that is 2n. Okay, so in the hexaploid, that our common wheat, or that is known as bread wheat or Triticum acetivum. Okay, so already discussed that this is the most widely cultivated species in the world. Okay, second is the spelt wheat, that is known as, that scientific name is Triticum spelta. Okay, so this is also a species that is largely replaced by the bread wheat. So this is a old type of bread wheat, but now the mostly bread wheat is the your Triticum acetivum. Okay, in the 21st century, this is optionally grown for the artisanal bread and pasta purpose. Okay, means common wheat is the Triticum acetivum. Now come to the tetraploids. So in the tetraploid there are three types, that is durum wheat, immer wheat, and horsan or kamut wheat. So durum wheat name is the Triticum durum, that is tetraploid, and this is also a widely used tetraploid and the second most widely cultivated wheat. After Triticum acetivum, Triticum durum is the mostly cultivated wheat. Then come to the immer wheat, that is known as Triticum turgidum, and subspecies is the Triticum dicoque. Okay, this is a species cultivated in the ancient times and derived from the wild immer Triticum dicoquoides, but now no longer in widespread use. Okay, so ancient time this is mostly tetraploid species. Then third one is the Khorasan or Kamut, that is known as Triticum turgidum subspecies Turenicum. Okay, also known as Triticum Turenicum. This is also ancient grain types, and this Khorasan is a historical region in the Afghanistan or the northeast of the Iran, and this grain is twice the size of modern wheat. मीन्स इसका जो ग्रेन साइज है वो आपका बहुत बड़ा होगा and have a rich nutty flavors. Okay, now next is the diploid species under this in corn, that is Triticum monococcum, which comes and that's it domesticated from the wild in corn Triticum voitinicum at the same time as a immer wheat. Okay, also there are hull and hullless wheat. So mostly ancient wheat are the hull type, means they are tightly wound and difficult to harvest in the threshing, means they are difficult to remove the grains. Okay, but nowadays most of variety are hullless. Okay, so this is about the major species. Okay, now come to the growth stages. Okay friend, so when we grow the wheat, there are subsequent so many stages, like first is the one shoot stage, second is the tillering stage, then tiller formed, then leaf sheath are strengthened, then leaf strongly erected. So this is known as tillering stage. Okay, and this is also very sensitive to the irrigation. Okay, critical stage for the irrigation. Then first node of the stem is arrows, second node is arrows, means it is a elongation phase or also known as stem extension. Then last leaf just visualize, means formation of flat leaf. Okay, then next is the booty stage. Okay, then there will be a next stage is the heading stage. So there is a formation of ear, head, then flowering, then there will be a ripening stage. So friend, there are so many stages in the growth cycle of the or life cycle of the wheat. So these are the important stages, like emergence, which is 0 days, then floral initiation, that is known as double digit stage, that comes under after 20 days in the spring types of wheat, and second is the winter wheat. So friend, this is also two types of wheat that is cultivated worldwide. That is, first is the winter wheat, that is mainly grow under the September, October month and harvested in the March. So this have high duration. And spring wheat mainly grown under the early spring, like February and March, and harvested in the late summer, that is your June to July. So this stage have less 4-month period. Okay, and this had approximately 6 months. So flower initiation in spring wheat take place in the 20 days, winter wheat 35 days, then terminal spikelet stage, it's come under 45 days, 60 days. First node appear in the 60, 80, heading 90, 120, anthesis 100, 130, physiological maturity at 170 days. Okay. So friend, this is about the different growth stages of the wheat crop, and these are the major crop, that is emergence, flower initiation, terminal spikelet, emergence of first node, heading, anthesis and physiological maturity. Okay. Now come to the some important traits. That is, first one is the plant height. So friends, you know that the ancient or old cultivars of the wheat crop are very long type, and they are up to more than 150 centimeter. But in this variety, there is a problem of lodging. So continuous breeding program targeted to reduce the plant height, and at recent time most of the variety or modern variety are dwarf or semi dwarf type. Okay, so this is very clear why we are talking about the plant height. So the suture of the stem is important in determination of cereal production, as it affects both yield potential and the lodging. Okay, so already discussed that if a variety having the high plant height, then it is susceptible to the lodging. Okay, so the semi-dwarf wheat are able to receive higher rate of fertilizers, means they are highly responsive to the fertilizer application because they are generally resistant to the lodging. Okay. Then breeding program throughout the world consistently reducing the stem height. Okay. And lodging is an important problem that reduce the yield by 30 to 40%, and reducing the plant height can improve this yield potential. Okay. So the optimum height in the modern variety ranges between the 70 to the 100 centimeter. So friend, this is the optimum height. Okay. Now what happened? The reducing now breeding for reducing further plant height is not suitable because already we received or we achieve the optimum plant height. Okay, so the lodging is independent of the stem height within a range of short plant, because we already have as optimum plant height. If we are reducing plant height below this, then there is a no effect on the lodging. Means if a variety having the 60 centimeter plant height or 80 centimeter plant height, then sensitivity to the lodging is almost equal. But if you are reducing the plant height continuously, then it affects the yield potential. Okay, because it reduces the biomass, is reduce the sink and the source tissue. So in this case, we can work about the stem diameter that is also sensitive to the lodging. Okay, so if we do breeding for the stem diameter, that is good for the development of lodging resistant variety. Okay, so this is about the plant height. Now come to the crop phenology. So friend, last to last slide, we have discussed that wheat have number of growth stages or the cycle, and each cycle have specific requirements in terms of resources and the environmental conditions. Okay, or temperature. This is a very important for the development of growth stages. Okay, so for this phenology, determine the adaptability of cereal to a fluctuating environment and affect yield potential. Means I want to say that if the period for the wheat growing, winter wheat is the late September to the early November. Okay, because there is a, after this there is a winter and there is a vernalization. At that time growth is slowed in case of winter wheat. Okay, if we change the sowing time, okay, if we change the sowing time, means if we grow the winter wheat in the late December, then there will be a no vernalization, and the temperature for particular stages is also changed, so it affects the yield potential. So the study of crop phenology, or you can say crop plasticity, is very important for the yield potential. So in wheat, length of period between the seedling emergence and the heading bar occasionally modified. Okay, so period of seedling emergence means vegetative phase is generally changed by the some photoperiod genes. Okay, or breeding approaches by crossing the long duration or short duration varieties, but the time to anthesis was not modified. Okay, however, in the less moisture area or drought prone areas, these phenological development traits are crucial for achieving terminal growth, because in the drought prone area mostly we need the drought escapers types of wheat which can complete the life cycle before the occurrence of drought or achieving the terminal drought. So when we change the crop phenology in the drought prone area, there is a positive results in the yield potential are observed. Like in Australian wheat, selection for the short duration varieties improves the yield. Okay, so the study of crop phenology is also important in wheat to improve the yield potential. Next is the dry matter production and the partitioning. Okay, so friends, you know that this is a plant and this is a leaf of wheat. Okay, this is the flower of wheat and this is a roots of wheat. Okay, so these are the photosynthesizing leaf, that is also known as source tissue, and these leaves are able to the photosynthesis and also responsible for the biomass production. Okay, and the inflorescence, even stem, root, grain, all are known as sink tissue. So these tissues are generally known photosynthesizing and they need this photosynthesis for their growth. Okay, so the distribution of this photosynthesis into the sink tissue known as assimilate partitioning. So this is also a very important topic, that what organ receive how much photosynthesis. Okay, so the yield improvement in newer varieties was more associated with the improved partitioning than with the total dry matter production. So our breeding approaches mostly targeting the more partitioning of this photosynthesis toward the grain. Okay. However, increase in grain yield were almost entirely independent of modification in biomass. Only there is a some exception. So therefore the partitioning is more dependent on it. Okay. So the intercept more radiation and radiation use efficiency and leaf area index were not much focused. Okay. Only this partitioning in the modern variety, the partitioning efficiency or assimilate partitioning is more focused, but the intercept more radiation radiation use efficiency and leaf area index were not much focused. Also the single leaf photosynthesis was not associated with the breeding program. However, maximum photosynthesis achieved by increase the GS, that is known as stomatal conductance. Okay. So there are some variety developed by the international maize and wheat improvement center, that is situated in the Mexico. Okay. So the simiate cultivars have high single leaf photosynthesis achieved by the increasing the stomatal conductance. So the increase in harvesting index, HI is the harvesting index associated with the high nitrogen use efficiency and short plant suture. Okay. So in the modern varieties, the harvesting index increased by the high nitrogen dose or nitrogen use efficiency or the dwarf variety. Okay, these traits are mostly avoided in the breeding program, that is radiation use efficiency, radiation interception, leaf area index, single leaf photosynthesis. Okay, however, this short suture is not beneficial under the drought conditions. Okay, because there is a no lodging. Okay, now come to the nest, that is known as yield components. So friend, that there are certain yield attributes like spike length, okay, spike size, then spike weight, effective number of tillers, then number of grain per square meter, per meter square, per meter. So they are even test weight, okay. So they are number of traits which are directly related to the yield. But the yield variation in wheat crop mostly focused in difference in number of grains per square meter than the individual grain weight. So test weight is mostly not focused, but grain number per square meter is mostly associated with the yield. In modern cultivar development program, the flow rate primordia just before anthesis increase the number of fertile flow rates. Okay. So friend, in the modern cultivars, there is a increase in the number of fertile flow rate which cause the higher yield. Okay. Also, the greater number of flow rate per spike at anthesis is more in semi-dwarf cultivars due to the reduced motility, because there is a less competition for the assimilates. Okay. Lower primordia between Flagley, Pepperens and anthesis. So there is a greater number of flow rate per spike. So these two factors increase the number of fertile flow rates, also flow rate per spike, responsible for the high yield in the modern cultivars. In semi-dwarf germ plant, there is a optimized plant height that is associated with improved harvesting index and the grain number. Okay. So there are two genes, that is RST1 and 2. This gene reduce the cell size in the above ground organs, inhibit the internodes elongation, improve partitioning to the reproductive part, and reduce flower abortion, because there is a more photosynthesis to the growing flowers. Okay. So that's why these two genes are responsible for booming the yield in the wheat, also responsible for the grain regulation. Okay. Now talk about the genetic basis of the yield improvement. So this semiat is the major center which are working on the wheat improvement program, and they expand the genetic base of modern variety using the conventional breeding, that is the crossing with the contrast parent, and the some cytogenetic approaches. And there is a introduction of some alien genes like 1VR. This gene transfer from the rye with the cross of Triticum torschi and Triticum turgidum. This gene present in more than 30 modern cultivars, and this increase the stress tolerance and also have good yield characteristics. Okay. Second, development of synthetic hexaploids with the wide crossing in between durum wheat and the wild diploids. This transfer the D genome and responsible for the disease resistant and good yield. And 50% of all the process of the semiat contain this D genome for synthetic hexaploids. Also the LR19 gene from the Agropyron elongatum improve the yield. So friend, these are the major genetic improvement program done in the wheat crop by the semiat. Okay. Now we'll talk about the physiological basis of the improving the yield. Okay. So there is a one scientist, that is Loomis and Amthor. They calculated that resource use efficiency, RU, is the resource use efficiency for wheat in the when they grow in the northwest Mexico. There is a total accumulated solar radiation is the 2050 million joule per meter square. Okay, for growth cycle. Okay, in the wheat growth cycle there is a 2050 mj per meter square. So this is the total solar radiation. Okay, while there is a canopy intercepted active absorption being 1750. Okay, also the quantum requirement is the 15 to 25 moles of photon per mole of CO2 fixed as a CH2 or carbohydrate. Okay, so the resource use efficiency varies in 1.5 to 2.6 CH2 million joule per. So this is very less compared to rice. Okay, generally in the wheat varieties it is near to 1.5 to 1.8, and the biomass vary from 2600 to 4550 gram per meter square. Okay, under irrigated conditions the cost of root growth and the maintenance for the 10% cost of above ground mass. So friends, you know that in the irrigated condition there is a good root growth and good maintenance of the root, so it can cost the 10% of the total above ground biomass. So biomass can be improved by the optimized plant height with improved intercept radiation, because only out of 2250, 1750 radiation are intercepted. Also we can improve the resource use efficiency. So 1.126, it can be increased up to 2. More than 2.5, and this can be achieved by the modification in photosynthetic metabolism, canopy architecture, and sourcing balance. Okay, so now talk about the radiation interceptions. Okay, so these are the leaves and these are the sunlight. Okay, so how the sunlight intercept these leaves and reach to the anothers. Okay, so the more radiation interception can be improved by increasing the length of growing cycle. So first strategy is that we can grow the total length of the growing cycles. Okay, so they receive the more light because there is an increase in the leaf area duration. Okay, our second approach is that we can increase the total daily interception by the canopy, or we can improve the canopy architecture. It improves the interception efficiency architecture. Okay, so the length of growing cycle can be increased by the photoperiod and vernalization genes. However, adaptation to the new growth cycle with environmental is also crucial. So first strategy is that increasing the length of growing cycle, which we can achieve by the change in the photoperiod and vernalization genes. But problem is that when we change the growing cycle, there will be a change in the G into E interactions, and also the strategy to adaptation to the new growth cycle, which may affect the yield potentials. Okay, so now we can go to the second strategy. So second strategy, cereal crop normally achieve maximum radiation interception before the onset of stem elongation. Okay, because at that time there is a maximum vegetative growth or the leaf area are the optimum. However, modification at this stage not effective at step not responsible to yield. Friend, stem elongation is the stage where there is a more leaf are available and we can achieve the maximum radiation interception during this period. But this phase is not directly responsive to the yield. So this phase is also not suitable for the, therefore suitable for short growing season and moderate severe water stress. But these types of strategy can be beneficial in the environment where is requirement of short growing season and moderate severe water stress environment. In this environment, these types of reducing the length of growing cycle is effective. Okay. Rapid canopy establishment crucial for the radiation interception and reduce the evapotranspiration. So resource use efficiency can be increased at critical stem elongation phase in this environment, and this can be improved the yield. Okay. Now come to the radiation use efficiency. So friend, already discussed that the radiation use efficiency is in the wheat crop is less potential. So a larger sink strength during post anthesis can improve number of grains. If we have more number of grains during the post anthesis phase, it improves the radiation use efficiency. Manipulation of leaf photosynthesis, mainly flag leaf photosynthesis, stomatal conductance, higher photosynthesis under stress, and nitrogen partitioning to the growing organ also can improve the yield. Then reducing photorespiration, because you know that C3 plants are 25% of the energy lost in the photorespiration process. So here is also a possibility to increase the photosynthetic efficiency. Also there distribution of incoming radiation, which can be modified by the size of the leaf, optimize the leaf size. All the acute leaf angle, not erect or not horizontal. This is acute leaf angle. Smaller and more erect upper cone means upper leaves are more erect and lower leaves are less erect. Okay. So by this way we can improve the radiation use efficiency. Now source sink balance. So this is also very important. Already discussed that what is source tissue and what is sink tissue. Okay. So the balance between these two are also crucial for the yield production. So after anthesis, source and sink both limited because there is a drying of the leaves. Okay. But sink are more limiting because this is the sink growing stage or they are in the log phase. So there will be a high growth. So they need more photoassimilate during the grain filling stage. So yield can be more subjected to the sink limitation. Okay. So already discussed that translocation of this gene improve the source sink balance. Okay. In a study of 6 NIL, that is known as near isogenic lines having the higher partitioning of assimilates to the developing spikes as anthesis increase the sink strength due to the larger spike. If there is a more transfer of assimilates toward the spikes are larger and they can accumulate or more assimilates to the spike. Okay. Also the lengthening of the spike phase is more promising because if there is a more lengthening of the spike phase, then more accumulation of photostimulates in the spikes, or it can improve the grain setting efficiency. So grain setting efficiency will improve the growth of carpels, lead to the greater average grain weight. So then this related to the grain weight or the test weight. So by this way, we can improve the salt sink balance. Okay. Next point is the water use and its efficiency. So Pesciora developed a model where yield is related to the water use, how much water is applied, how much efficiently it utilized, and the harvesting index. So phenology is also important, which related to the water use. Okay. Deeper root system, early vigor, this reduce the evapotranspiration. Okay. Also lengthening of the spike phase is more promising because this phase is more water use efficient than the stem elongation. Okay. So by this way we can improve the water use efficiency. Then there are certain physiological tools like eliminating inferior agronomic phenotype visually in the early generations. So if we can remove the inferior phenotypes in the earlier stage, it can improve the yield. Selecting superior physiological phenotype using rapid detection techniques like SPAD, okay, thermal gun in intermediate generations, and selecting for higher yield performance in advanced generation by the yield drive. So these approaches can improve the yield performance. Okay. So there are some spectroradiometrical indices. So you know that there is a spectral reflectance. You know that there is a differential spectral absorption and reflection of the soil and the plant organ. So this is the sunlight, and there is a absorption and reflection is difference in the soil and the plant organ. So red light and near infrared light, there is a near infrared light reflection, and this can be used for the this spectrometric use can for the estimation of leaf area index, radiation interception, canopy chlorophyll content, xanthophyll cycles, photochemical reflectance index, that is PRI. Okay. So for this we use NDVI. NDVI is the normalized difference vegetation index. So friend, this is a NDVI sensor which is developed by the NASA, and it uses the red and near infrared reflectance which can use to estimate these traits, and also can be used for the resource use efficiency. So this instrument used for the rapid detection or screening of the physiological traits. Now second trait is the that is known as thermal gun. Okay. ठीक है. This used for the measurement of canopy temperature. Okay. So canopy temperature depression CTD is measured with the infrared thermometer or the thermal gun. So friend, when the water status is reduced, okay, if there is a less water, then there will be a closing of the stomata. Okay, it causes the increase in the canopy temperature because there will be a lack of transpiration. Okay, तो क्या होगा जब आपका water stress आएगा, there will be a closing of the stomata. If there will be a closing of stomata, there will be a less transpiration cooling because transpiration कम हो रहा है तो आपका cooling effect भी कम होगा, तो वहां पर क्या होगा, there will be an increase in the canopy temperature. So we can measure by the help of this infrared thermometer, and this can be used to develop the crop water stress index, and this instrument can be used for the drought resistance mechanism or avoidance. When midday there will be a lower canopy temperature has so better water stress. If exemplar having a lower canopy temperature during the midday means this plant is transpiring better. Okay, Canopy temperature depletion and yield best expressed under the when there will be a well water, high evaporative demands. So heat stress and drought stress genotype can be screened by the help of this canopy temperature meter. Okay. So friends, earlier we have discussed the NDVI and the canopy temperature. These are the used for the instantaneous or there is a infrared gas analyzer, infrared gas analyzer. So these instruments are used for the instantaneous measurements. Means हम अगर आज का measurement ले रहे हैं तो आज का. They are not useful for the whole growth stage. So for this we use the stable measurements, that is known as carbon isotopic ratio, okay, or CID, carbon isotopic determination. So friends, you know that in the atmosphere, carbon dioxide that is responsible for the photosynthetic process contain the 98.9% is the C12, that is normal carbon, and that 1.1% having the C13, that is the isotopes of this carbon. And there will be leaves which discriminate against the C3 and laser the ratio of C3 to C12 means less assimilation of C13. So there is a one instrument that is known as IRMS, isotopic ratio mass spectrophotometer, used for the measurement of carbon isotopic discrimination, and this is the control we use, that is -8. Also there is a two enzymes, that is Rubisco and Pepcase, and this is a major enzyme which fix the carbon in the C3 plants, and Pepcase which fix in the C4 plant. So capacity of these two enzymes is also different for the discrimination of this C12 and C13. So this factor is the 4.4. Okay. Also, if a plant showing the higher delta 13C value, means there will be a closing of the stomata. So by the help of this carbon isotopic discrimination, we can measure the stable change in the genotype, and also we can screen for the drought and stress tolerance. Okay. Also, Kimmiat cements coined the term mega environment for the study of the genotype into environment interactions. So friends, you know that when we go for the breeding strategies or breeding experiments, there is a large number of germ plant. It could be more than 1,000 or more than 4,000. So the study of these germ plant into the field condition and under different abiotic conditions is difficult. Okay. So it is very difficult to test such number kind of germ plant. Okay, or large genotype and environment interaction. So mega environment is used for the selection of trait for control and test environment in the breeding trials. So if we develop this mega environment, we can understand the genotype and environment interaction for development of stress tolerant varieties. Okay. Then there will be a development and application of molecular markers, the marker state selection for predicting and tracking of the valuable alleles, the analysis of genetic control of specific traits, because these stress are the QTLs, quantitative trait loci, regulated by the number of alleles or by the analysis of the whole genome. Okay. In breeding, we can detect the origin of specific allele introduced from the Binder process or the gene for vernalization response, fold tolerance have been located in chromosome of homologous group 5 like BRNA1 locus, chain shortening the life cycle in the wheat transfer by the biotechnological ways. Also QTLs for the water use efficiency, yield traits such grain weight, ear compactness, lodging resistance, heading time, abiotic stresses such as drought, heat, salinity. Identification of this QTL can use for the yield improvements and the novel approaches such as integrated omics that consider the genomics, phenomics, ionomics, okay, then transcriptomics, and the new approaches that gene editing, that is CRISPR-Cas9. These advanced technology can help in the identification of valuable alleles or the traits which are crucial for the yield improvement. So friends, this is about the wheat crop physiological breeding and biotechnological approach. I hope you understand this lecture. Thank you very much.